So let's practically look at this paper and be able to theoretically introduce some concepts to you where necessary. And it is a paper that was brought to some students when they were having their end year examinations. And the first question reads, give two unique features of members of Kingdom Monera. So this is biology. And of course, everybody knows that we have five main types of kingdoms in as far as classification of organisms is concerned. We always have this first kingdom called Kingdom Animalia that everyone knows more about. We have another kingdom called Kingdom Plantae, and another one is called Kingdom Fungi. And then we also have another kingdom that we always refer to as Kingdom Monera, that is here. Then we have Kingdom Protoctista. Kingdom Protoctista, or you can as well be able to call it Kingdom Protista. So I've actually done much of the classification of this kingdom called Kingdom Animalia, and I may not actually be able to say more about that kingdom. And maybe I've also done this in as far as the classification of Kingdom Plantae is concerned. I've not done much about the Kingdom Fungi, and I'll be able to give you some information concerning that. But of course, I just said Kingdom Animalia can actually be able to get further classified under several phyla. And I did mention about phylum Chordata or Chordata. We have phylum Arthropoda. You see, those are just the examples of phyla that you can be able to actually find under Kingdom Animalia, including phylum Nematoda that houses the tapeworms, and phylum Annelida that actually has a very good example like the earthworms. We may have other examples of phyla under Kingdom Animalia like Vocifera, Echidnomata. You see, those are just examples of, or even Platyhelminthes. So we can actually be able to mention as many as we can, but majorly you should know this. Kingdom Animalia is mainly classified into 9 phyla that can further be able to get discussed at a high level of learning if someone does much of what you call zoology in the university or in the higher learning institutions. Kingdom Plantae can be able to get further classified into 3 very important divisions. We have division Bryophyta. Remember, in as far as the classification of Kingdom Animalia is concerned, we do always talk about phyla. So from kingdom to phyla in the classification of Kingdom Animalia. But in the classification of Kingdom Plantae, we do always talk about divisions. So I'll talk about divisions, not phyla. So division Bryophyta, we have division Pteridophyta, we have what you call division Spermatophyta. We'll get time and discuss more about this classification. But of course, everybody knows division Bryophyta consists of examples such as mosses and liverworts as plants under that classification. They are non-flowering with a particular organization of simple leaf-like and stem-like structures. And they actually tend to have the male parts and the female parts in terms of the production or the actually making of gametes. We have what we call the antheridia as the male parts and archegonia as the female parts. And of course, they have divisions, and that can actually get further discussed later. And you see, you can also be able to talk about this Pteridophyta as our second division, and this encompasses examples like ferns and horsetails. They actually lie under that category. They also have divisions, and that means you can further be able to discuss those subdivisions under that division later. And you see, you can actually be able to actually mention these funds and hostels as also structures with certain features. Of course, like a fund plant has a compound leaf, or fund plants have compound leaves that have leaflets called pinna, and they have spore producing structures just underneath the leaflets. And those spore producing structures organize themselves or are able to actually get put in groups called sori or singular sorus. They have rhizoids, of course, to anchor them to the ground and be able to enable them absorb water and mineral salts. And most of these plants, including even that division called division bryophyta, do not have a well-developed vascular bundle consisting of the xylem and the phloem. And literally, you might actually realize that bryophyta may have uh, the gametophyte generation being dominant and the sporophyte being born on that gametophyte and being dependent on that gametophyte in terms of the alternation of generations. Then the pteridophyta may actually have the sporophyte being dominant and the gametophyte being smaller, very reduced and enclosed within the sporophyte. You can look at that. And literally, if you look at the spermatophyte as our last division, it consists of the flowering plants. It has two very important subdivisions. Those divisions are actually given examples such as gymnospermaphyta and angiospermaphyta. And you can have the angiospermaphyta getting classified further into classes. So you have actually the kingdom, the divisions, the subdivisions, then now the classes. So the kingdom, kingdom plantae. The divisions, bryophyta, pteridophyta, and then spermatophyta. The subdivisions of spermatophyta, gymnospermaphyta, and angiospermaphyta. The classes of angiospermaphyta, we have what you call monocotyledonae, that will consist of all the monocots with one cotyledon in terms of the way their seeds look like. They also have what you call a fibrous root system. Their leaves are narrow, long, with parallel venation. And of course, if you look at their structures, you realize that they do not have anything like secondary growth. Their stems do not increase in width or girth. Why? Because they lack what you call the cambium ring. You see, and that absence can actually make them not to develop what you call secondary growth. And we have what you call dicotyledonae. Dicotyledonae. This consists of dicots, and of course, examples of monocots include maize, rice, grass, sorghum, millet, wheat. Dicotyledonae as another class consists of all the dicots like the beans, the peas, the mango tree, and of course any plant that has one network or reticulate venation should fall under that category. 2. Having a taproot system. Having a cambium ring that can facilitate secondary growth, making these plants to increase in their width or girth in terms of their stems. And of course having two cotyledons. And of course, if you compare between the mono and the di, you realize that the arrangement of their petals will differ. This will have their petals being in threes of their multiples, and the other one has their petals being in fours, fives, or their multiples. You see, so if you look at the gymnospermaphyta, the classes are three. We have what we call the cycadales. These cycadales are the ones that will have a resemblance to the palm trees. We have what you call coniferales. These will majorly be able to consist of the cones, and you have what you call the ginkgo biloba, ginkgo biloba, ginkgo biloba. So that can actually be able to give us those three very important. So this subdivision called gymnospermaphyta under a division spermatophyta of kingdom plantae will consist of three classes: cycadales, whose appearance will resemble the palm trees; Conference consisting of the cones. Cones can actually be able to survive at an altitude of 2,000 meters above the sea level. And they have two types of the cones in terms of the development. One you have what you call the male cone, and you have what you call the female cone. The male cone will actually be able to occur at the terminal part of this plant, or will be born on the terminal bud of this plant. Then, of course, the female cone will actually be able to branch from the stem, laterally develop. And the female cone will actually be able to have seeds getting born on it. And the female cone is normally larger than the male cone. Ginkgo biloba is a very good example. And you see, this can actually be able to have a plant like the ginkgos, you see, or the ginkgos being the class and the ginkgo biloba being a very good example. So you can talk about the ginkgos or the ginkgo biloba. The ginkgo biloba becomes an example. The ginkgos becomes the class. And literally, this can majorly be able to get found in China. They have the female and the male parts, of course, but on the same plant, you see. And their leaves are funnel shaped, you see. So those are just some of the features you can be able to capture as far as that classification is concerned. Kingdom fungi, of course, consists of almost all the non-green plants or structures or organisms within our environment. You can talk about the mushroom, you can talk about the yeast, you can talk about the toadstool, you can talk about the puffball. So those are examples of non-flowering organisms that can fall under that kingdom called kingdom fungi. So getting back to kingdom monera consists of all the bacteria in our environment. And of course, this kingdom called kingdom monera will have unique features as per the question. One, when all these other kingdoms are eukaryotic, to mean the nucleus that contains the hereditary material that can get transmitted from the parent to the offspring is membrane bound. But in as far as kingdom monera is concerned, all the bacteria have their nucleus containing the hereditary material or genetic material being not membrane bound. So they are prokaryotic. They are prokaryotic. They are prokaryotic. So members of kingdom monera are prokaryotic, to mean their nucleus is not membrane bound. They have a naked nucleus containing the genetic material. The second feature that seems to be very unique to this category is they actually lack very many major organelles. We said an organelle is any structure found in a cell. So most of these members have to actually end up lacking very, very, very important organelles. Like they lack the mitochondria, you see. So that tells you that their type of respiration should be anaerobic. That means they can break down whatever is actually inside their structure in the absence of oxygen. So they lack major organelles within their structures, such as the mitochondria, the plasmids, the Golgi bodies, and others. So lack major organelles. Lack major organelles. And you can also be able to mention about them being anaerobic majorly and some being aerobic. So some can respire, or a good number can respire anaerobically, while some can respire aerobically. Respiring in the presence of oxygen, respiring in the absence of oxygen. These members are either autotrophic or heterotrophic. Autotrophic or heterotrophic. That tells you that some contain the green coloring matter called chlorophyll, so they can photosynthesize, they can make their own food in the presence of sunlight. Look at that. That means they can actually feed on or obtain nutrients from what they've actually manufactured. But being heterotrophic mean they do not have the green coloring matter, and the absence of this can make them to rely on already manufactured food saprophytically, where they feed on dead decaying matter causing decomposition. And you can also expect us to mention about symbiosis, where the bacteria can live in the intestines of some animals. Look at this example. We are told that bacteria live in the intestines of human beings. They help in the making or in the synthesis of vitamins K. Look at that. And in the end, or in return, human beings provide shelter and nutrients to this bacteria. Go back to the other category. Some bacteria do find shelter in the intestines, you see, or in the alimentary canal of animals like the cows, which are ruminants. And in the process, these bacteria in these ruminants help in the secretion of an enzyme called cellulase. Cellulase helps in the breakdown of cellulose that animals feed on to actually form sugars that are directly utilized by these animals. Try to imagine a bacterium is living in the stomach of a cow. In that stomach, that bacterium is able to access shelter and nutrients from this cow. And in return, the bacteria ensures that enzyme cellulase is produced to break down the cellulose that makes up the cell wall of the plant cells that these animals feed on. Because ruminants feed on vegetation. Vegetation consists of plants, of course. The plants have cells. The cells that make up the plants are made up of cell wall. And the cell wall consists of the cellulose. Look at that. The cellulose has to get digested. How? By the enzyme cellulase. That its secretion relies on the presence of this bacteria. That's why in human beings like us, we do not break down the cellulose. Why? Because we do not have the bacteria in our intestine that can actually help us to secrete enzyme cellulase to break down the cellulose that makes up the cell wall of plant cells. Yeah, and of course you can mention so many features of this kingdom. Other features including they are unicellular or single-celled. They are made up of a single cell. That tells you they are microscopic. You see, they are microscopic. You see, they can actually be able to exist as single organisms, or they can form what you call a colony. You see, they're assembling together. You see, and we have so many types of bacteria in our environment: Streptococcus, Staphylococcus, Lactobacillus, Nitrobacter, Nitrosomonas, Nitrococcus. They are very many. They are very many. Azotobacter, Nitrobacter, you see. So they are very many. They are very, very Diobacillus, Pseudomonas, you see. We have another one called Treponema pallidum, Vibrio cholerae, Salmonella typhi. Those are examples of bacteria that will actually be able to put in this category. Now let's just get into the second question. And this question says, the diagram shown below represents a cell. Identify two reasons why this is a plant cell. And of course, if I look at this diagram, I can actually be able to realize that we have both a plant cell and an animal cell. We always say that plant cells and animal cells can either get viewed under a light microscope or an electron microscope. We're always told that whenever a light microscope is used, and on the other hand, an electron microscope gets used, the end product in terms of the images you're actually going to form cannot be the same. Why? Because one, a light microscope has a lower magnification of relatively 1,000, and an electron microscope has a higher magnification of around 500,000 times. So that means a light microscope can actually enlarge something up to 1,000 times, while an electron microscope can magnify something up to 500,000 times. I also told that a light microscope has a lower resolving power. That means when a light microscope is used to view a specimen, it cannot make the parts of that specimen very distinct from each other. It cannot bring out that aspect of differentiation. But when an electron microscope is used, it can clearly make each and every part of the specimen distinct from each other. So clarity is brought out when an electron microscope is used. Why? Because an electron microscope has a higher resolving power. A light microscope can only view dead and live specimens, but the use of an electron microscope restricts on the viewing of only dead specimens. Look at that. We've actually been told that a light microscope can actually be able to make use of objective lenses, while an electron microscope can maximize on the use of electromagnetic lenses. A light microscope may require these simple staining what? Substances like iodine, but an electron microscope can demand the use of very complex staining what? Pigments made of very strong metallic what? Elements. So regardless of what you're going to use as your microscope, we should actually be able to bring out the differences between a plant cell and an animal cell. How? One, we should know this, that a plant cell has both a cell membrane and a cell wall. And this is our cell membrane, and this is our cell wall. So the first thing that makes this to be a plant cell is it has both a cell membrane and a cell wall, while an animal cell will only be able to have what you call a cell membrane. That's the first difference. Number two, you'll realize that the cytoplasm and the nucleus in an animal cell are centrally located. But the nucleus and even the vacuole of this plant cell can actually have distinctive positions. How? I've actually to repeat this. One, the nucleus and the cytoplasm of the animal cell is centrally able to get position in this cell. But now, if you look at the nucleus and the cytoplasm of this plant cell, they are pushed towards the periphery. Have a look at that. So, the nucleus and the cytoplasm are centrally positioned. The nucleus and the cytoplasm are pushed towards the periphery. Okay, something else to note. The vacuole in a plant cell is large and dominant. Vacuoles in animal cells are absent, but if they are present, they are small structures just scattered within the cell. A vacuole in a plant cell is large, dominant, and centrally positioned. A vacuole in an animal cell is absent, but when present, it can just be a small structure scattered within this cell. Now, a plant cell is normally regular in shape. I'll just not even just consider that in my drawing, but you realize that an animal cell is irregular in shape. Don't mind about the diagrams. Take note of the points I've actually to mention out. I've said a plant cell is regular in shape, but an animal cell is normally irregular in shape. Look at that. A plant cell is larger. Forget about the sizes at this moment. A plant cell is normally larger. An animal cell is often smaller. A plant cell is normally larger. An animal cell is often smaller. That is what you can be able to give as another difference. So we've actually mentioned the differences, and we are good to go to the next question. Name the organelle found in high number in the following structures. I've actually told you an organelle is any structure you can be able to find in a cell. So we expect these ones to be cells. So a palisade cell forms part of what you call the palisade mesophyll tissue. Because I told you, a tissue is made up of several cells which can get coordinated to realize a particular function. So, a palisade mesophyll tissue consists of palisade cells. They are found in a leaf, in a plant. If you look at the structure of a leaf internally, from the top, we have what you call the cuticle. Just below the cuticle, you have what you call the upper epidermis. Just below the upper epidermis, you have what you call the palisade mesophyll tissue. Below the palisade mesophyll tissue, the spongy mesophyll tissue. Within that region, you have what you call the vascular bundle tissue or the conducting tissue. that consists of the xylem and the phloem. We know very well the xylem transports water and dissolved mineral salts from the roots to the leaves, while the phloem translocates manufactured food from the leaves to other parts of the plant. Just below the spongy mesophyll tissue and the vascular bundle, just within the same region, consisting of the xylem and the phloem, we have what we call the lower epidermis, that consists of what we call the gut cells that control the opening and the closing of the stomata. So, palisade mesophyll tissue consists of palisade mesophyll cells. These cells have an abundance of the chloroplasts as organelles. These chloroplasts contain the green coloring matter called chlorophyll, and the chlorophyll traps light energy from the sun to facilitate the making of food in these plants. And of course, plants are autotrophic. A good number of plants, most plants are autotrophic. They actually make their own food and do not rely on any other source in as far as the requirement of what they may have as food is concerned. So they contain chloroplasts in large numbers containing chlorophyll that trap light energy to help them in making food. So they are actually autotrophic. So they are autotrophs. Autotrophic as a mode of feeding, and then themselves are actually autotrophs. And of course, someone can ask you to give the features they have that can make them to maximize on the making of food. One, their arrangement and position on the upper part of the leaf will enable them to actually absorb maximum sunlight for photosynthesis. Remember, the upper part of a leaf is greener than the lower part, to mean that the upper part is made up of majorly the palisade mesophyll cells, having large numbers of chloroplasts with many chlorophyll molecules, making the upper part of the leaf greener than the lower. And of course, they have large numbers of chloroplasts, as I've actually said, as another adaptation feature that will actually contain many chlorophyll molecules, and that increases the absorption of light and the manufacturing of food in the plants. And of course, apart from the palisade mesophyll cells, which other cells can also manufacture food in the leaf? One, the spongy mesophyll will also contain chloroplasts, although few in numbers, and as well the gut cells in the lower epidermis. They also contain chloroplasts that can help the plants to manufacture food. Now, goblet cells. Goblet cells are the cells found along the breathing and digestive systems. When you go to the trachea, you'll find the goblet cells. When you go to the alimentary canal in human beings, for example, you'll find the goblet cells along the lining of the stomach, the goblet cells in the small intestine, and these cells ensure that they secrete mucus. The mucus in the digestive system will lubricate the food and lubricate the lining of the alimentary canal to facilitate peristalsis to occur without any difficulty. Remember, the food has to move from the mouth to the esophagus, down to the stomach via the cardiac sphincter at the opening of the stomach, then from the stomach, exiting the stomach via the pyloric sphincter to the duodenum, the upper part of the small intestine. From the duodenum to the lower part of the small intestine, the ileum, and then later on, whatever is very useful as the end product of digestion gets absorbed in the bloodstream. Whatever remains as the undigested and the indigestible can get removed by the anus to the outside, and that's what we call agitation. The removal of the undigestion, agitation, from the word exit. The removal of the undigested and the indigestible material. That's what we call agitation. So these cells need to secrete mucus. We need to ask ourselves, which organelle engages in the packaging, in the transportation and secretion of what the cell may require? That is Golgi bodies. Or Golgi apparatus. So Golgi body or Golgi apparatus will be an organelle that will be abundant in the Golgi oriented cells called the goblet cells, so that there will be an easier manufacturing or synthesizing and secretion of mucus by these cells. When you go to the trachea, you'll find the goblet cells. They're along the lining of the trachea. They secrete mucus. The mucus traps the dust and the bacteria that may want to find their way into the what? The small intestine to cause infections. So When the cilia or the hairs along the trachea beat in waves, they push this mucus towards the opening of the trachea or windpipe. So that as the mucus moves towards the opening of the trachea or windpipe, it is moving along with the dust and the bacteria, which might have found their way into the trachea or windpipe. Now, we have different types of muscles. We have the biceps, we have the triceps forming part of what you call the forearm, we have what you call the trapezius muscles on your back, you see, and all these muscles, you have what you call the skeletal muscles forming part of what you call the hind limbs. These muscles have to have what you call mitochondria in large numbers. I'll explain why mitochondria. So mitochondrion, if it is one, and mitochondria in plural. Now look at this. These muscles, they are either skeletal or not. And of course, we have different types of muscles. We'll discuss this later. We have what you call the cardiac muscles forming what you call the heart. You have what you call the skeletal muscles. Look at this. The epithelial muscles. All these muscles have mitochondria in large numbers, with an exception of some. It may have either few mitochondria or mitochondria in small numbers, literally. But skeletal muscles have large numbers of mitochondria. Why? The mitochondria provide a site for respiration so that energy can get yielded, and that facilitates the contraction and the relaxation to bring about movement, because these muscles are able to facilitate the contraction and the relaxation. If they get triggered, and at the end of the day, they actually bring about movement, which results into the change in position by a mechanism. So I walking from this place to the market is facilitated by what you call the skeletal muscles in my hind limbs. Those muscles must have a large number of mitochondria where respiration is going to occur for them to get energy to contract and relax to bring about movement. So mitochondria should be in large numbers to actually provide a site for respiration so that enough energy is generated for the contraction and relaxation to bring about movement. Now, next question. Account for the following observations. A plant cell does not burst when placed in hypotonic solution. So if I take this to be my container, for example, and I have this as my plant cell, just look at this. You see, and you're told the surrounding is hypotonic, to mean the vacuole of this plant cell is hypertonic. So the surrounding of the plant cell is hypotonic. And the cell sap of this plant cell is hypertonic. And you see, this borrows knowledge from what you call osmosis as a physiological process. What I need to tell you is this. When the surrounding is hypotonic and the cell sap of this plant cell is hypertonic, then that means water or solvent molecules are going to move from a hypotonic region or from a lowly concentrated region to a highly concentrated region or a hypertonic region in the cell through osmosis across the semi-permeable membrane. And at the end of the day, what happens? The cell begins to have its vacuole increasing in size. And as the vacuole takes in more water, it starts to distend, it starts to increase in size, and it begins to exert a particular pressure on the cell wall. The pressure the vacuole exerts on the cell wall is called Tagger pressure. What do you expect if it is on an animal cell for it to expand or to extend to a particular level or point, then be able to burst? But a plant cell cannot burst. Why? Because as the vacuole takes in more water and then expands or increases in size, it exerts a pressure called Tagger pressure on the cell wall. To prevent the plant cell from bursting, the cell wall has to actually develop a pressure that it can actually use to withstand the pressure exerted onto it by the vacuole. And the pressure that the cell wall develops is called wall pressure. So the wall pressure prevents the Tagger pressure from causing the plant cell to burst. So as the vacuole expands and extends and increases in size, it exerts Tagger pressure on the cell wall. The cell wall develops an equal and opposite pressure called wall pressure to prevent any further expanding of the plant cell, preventing the plant cell from bursting. So your answer is a plant cell has a rigid outer covering called a cell wall that prevents it from bursting when it takes in water from a hypotonic environment. The vacuole takes in water because it is hypertonic to the surrounding. And that means it expands, exerting tugger pressure on the cell wall. To prevent this cell from bursting, the cell wall develops an equal and opposite pressure called wall pressure. And that pressure prevents any further expansion of this cell due to the intake of water, preventing this plant cell from bursting. Why does an amoeba not end up bursting when placed in distilled water? An amoeba falls under a kingdom called kingdom Protoctista. There's a kingdom called kingdom Protoctista. Kingdom Protoctista. Protoctista. And this kingdom consists of two subclassifications. One, we have what you call protozoa, and we have what you call algae. You see, protozoa are animal-like organisms under this kingdom. Algae are plant-like organisms under this kingdom. So examples of animal-like are like the amoeba, what we call trypanosome, what we call Vorticella, and many others. You see, and algae could actually consist of Chlamydomonas. They are what we call the chloroplast, and that means they can be able to manufacture food as far as this kingdom is concerned. Chlamydomonas. Okay, Chlamydomonas, you have what you call the Euglena, you have what you call the Spirogyra, you have what you call the Sargassum, you have what you call the diatoms. These are examples of the algae looking like or members under the subdivision algae in as far as the classification of kingdom Protoctista is concerned. The other one is actually amoeba, trypanosome, vorticella, what you call paramecium, what you call the plasmodium. And sometimes it's very difficult for us to even classify Chlamydomonas. Why? Chlamydomonas can look like a plant because it has what we call the chloroplast. And on the other hand, it has what we call the locomotor structure that's a feature in animals called the flagella. Look at that. Look at the Euglena. It has a chloroplast that can make it to manufacture food. So it looks like a plant. And on the other hand, it has an important what? Locomotor structure called flagella that can make it to move around. An amoeba does not have any chloroplast. An amoeba has a membrane-like appearance. It is actually unicellular, and of course, it does not have cilia or flagella as locomotor structures, but it uses what we call pseudopodia. 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 So let me look for a pen that can help me to finish this. So pseudopodia. Okay. So pseudopodia that can be able to help it move. Of course, pseudopodia are just the adjustment of the cell membrane in terms of being able to form particular shapes to facilitate it from moving from one place to another in the water. And of course, it has two very important what? Vacuoles. It will be called the food vacuole and the contractile vacuole. So the contractile vacuole, then food vacuole. So as it takes in more water, since it has been placed in distilled water, all the excess water accumulates in the contractile vacuole and not in the structure of this organism. So once this water has accumulated in this contractile vacuole, the vacuole moves closer to the cell membrane, then bursts, releasing the excess water to the surrounding. So there's no way in which this organism can again burst. It cannot burst. Why? Because it has a contractile vacuole that accumulates excess water in its structure. The vacuole moves towards the cell membrane, then bursts, releasing the excess water to the surrounding. So that protects from this amoeba from bursting when placed in distilled water. So this contractile vacuole is very important in osmoregulation. And of course, the work of the food vacuole is to actually ensure that the food within the amoeba is stored, then digested, so that this amoeba can be able to get nutrients. And an amoeba has only a cell membrane. Yeah. Of course, the paramecium uses cilia to move around the environment. So cilia and flagella are locomotor structures found in this kingdom, and those structures are made by an organelle called centrioles. The centrioles are responsible for the making or the synthesis of cilia and flagella, which are locomotory structures. Thank you for your time, and let's meet in the next lesson.